Good morning or afternoon, everyone, and thank you very much for coming along to this webinar for Epigeum's new course, Research Quality and Reproducibility, ensuring that researchers are engaging in responsible conduct. We should take about 45 minutes of your time, including the designated question and answer session. If you are familiar with this GoToMeeting software, you know there is a question option as part of the panel on the right-hand side of the screen near the bottom. If you would like to ask questions as they occur to you, please feel free to send them over and we will answer them at the end of our webinar. We are recording this webinar, so if you wish to view it again or to share it with your colleagues, it will be available on Epigeum's YouTube page next week. At the moment, everyone is on mute in order to avoid background noise. But again, if you have any questions, please feel free to ask them on the bottom panel. Just a couple of introductions. That's me, Anne-Marie Turner, on the left-hand side. I am the marketing manager for Epigeum's research courses. Just as a quick aside, throughout the webinar, we will commonly refer to this course as RQR. With that, I would like to turn it over to our two panelists, Dr. Rebecca Davies and Sarah Andrus. Dr. Rebecca Davies, would you like to introduce yourself?
Hello, everybody. Yes, this is Rebecca Davies from the University of Minnesota. It's so nice to be with you, and I appreciate your attendance at this webinar. I'm an associate professor at the University of Minnesota, where my interests are comparative animal endocrinology and veterinary diagnostic medicine. Although for the past probably 5 or 6 years, I've become very focused as the director of a new program called Quality Central, where my team works to help researchers and other scientists integrate best practices associated with research quality assurance into their research environment as a way primarily to support and help them demonstrate their ability to conduct rigorous and reproducible research. So I'm interested in this because research quality assurance skills are very rarely taught in basic research settings, and I think that they provide our scientists with an important tool that could be used to enhance and promote the quality of their work. So I've been working individually to create research quality assurance training programs for our college, but I really see a strong and great need for a standardized, flexible, convenient, and well-tested program that could be adopted by many scientists and many institutions. And that's why I became very interested in Epigeum and its approach to the development and evaluation of research training content. And I especially admire their consultative model where they plan to work with many scientists to kind of hit the sweet spot with this training material. So I really look forward to participating as a lead advisor for the Research Quality and Reproducibility course.
Thank you so much, Dr. Davies. Sarah, would you like to introduce yourself?
Hello, I'm Sarah Andrus, and I am the commissioning editor at Epigeum, managing the development of this program, Research Quality and Reproducibility. And I've been working very closely with Dr. Davies as well as our other collaborators in the US as well as the UK and Australia and New Zealand in order to bring this project to fruition. And so I look forward to talking a bit more in my presentation about how we're going to do that.
Thank you so much, Sarah. First, I will provide a bit of background on Epigeum itself. Next, Rebecca will have a presentation on the background and the vision of our Research Quality and Reproducibility course. Then Sarah will present an overview and discuss how Epigeum develops courses in general. And finally, we will wrap with a question and answer session. So to begin, Epigeum was founded in 2005 as a spin-off of the Imperial College London. Our two founders created the company in response to what they felt was a lack of timely support for researchers. At the time, one of our founders was getting his PhD while also working full-time when he came across issues that he wanted and needed to be advised on. The courses that he needed to take were either face-to-face classroom courses that would start over 3 months after he needed them, or he would find online material that was dated or inadequate. He saw this as an opportunity to work with universities to create supportive materials for researchers, particularly PhD students and postdocs like himself, that gave immediate support for all those issues. And that's where Epigeum started. In the more than 10 years since, we have expanded with our team from our first study program for students to also include research, teaching, and leadership and management courses intended for staff and professionals. Epigeum was acquired by Oxford University Press in May 2015. Since OUP is a division of Oxford University, Epigeum is still a not-for-profit organization in a higher education context and still very much focused on creating high-quality materials. To date, in the decades since we have been formed, we've created over 92 online courses across 19 different programs, and we currently have 4 in development, including RQR. We're also a globally focused organization, and we have worked with over 280 universities across 30 different countries worldwide to date. I would like to touch briefly on implementation, which is how the course can be used at your institution. Firstly, it can be used as a standalone academic course that you can supply to the students with an online, multimedia-rich course immediately at the time and place that they need it. This would be what we call a self-study mode. The advantage here is that you can make the course quite personal and release training on demand, so this is the most flexible option of the 3. Secondly, we have the option for tutor-supported online courses. There is plenty of supplementary materials available as part of each course, which can be used by tutors to enhance the course. Tutors can be used to provide feedback that cannot be provided by computers. They can work on community-based features, give motivational messages, and so there's a whole tutoring side to the course that can only be used if you have the tutors. The advantage here is that the course is allowed to be more effective, but you lose flexibility as you would tend to have fixed start and end times. Finally, the most common use for our course is part of a blended learning program. Typically how this works is that the students are given access to the online course ahead of a workshop, and then the workshop would really focus on consolidation and application of the material. This is considered to be maybe the most effective pedagogically, but it's also the least flexible. Thus, you have a spectrum here from the most flexible to the most pedagogically effective. So you can implement the course in all 3 modes at once. So together, these are the 3 methods of implementation provided, and they provide you with quite the armory to get your students the support and knowledge they require. Now that we have a bit of background on Epigeum and how we put our programs together, I'd like to turn it over to Rebecca and have her present the vision for this new course. Dr. Davies?
Thank you, Anne-Marie. Okay, the key objective for the Research Quality and Reproducibility course is to ensure that researchers develop the skills that are required to plan and execute well-managed, rigorous, and reconstructable scientific research, with a focus always being on achieving outcomes that are reproducible. The hope for this proposal, this project, is to support that skill development throughout the career arc of the researcher. So the goal is to provide training that's tailored to meet the scientists where they are as they progress through their careers, from undergraduates all the way through to scientific leaders. This slide illustrates that progression dependent on whether the scientist is working in an academic research lab or involved in corporate commercial research. We still have that progression from the undergraduate, as you see here on the slide, all the way through graduate student, postdoc, staff scientists, to the point of becoming principal investigators and eventually research administrators or research directors. And as you can see on the bottom part of the slide, we have this progression also in skills development that build on the skills that they learned previously. And the course structure Epigeum is proposing includes that additive approach of new skills that are required throughout the arc of the researcher. This slide comes from a commentary published in Nature in May 2016 this year, where researchers were given the opportunity to answer some survey questions. And as you can see in this slide, there's a response of about 1,500 researchers who were asked the question of whether or not there was a reproducibility crisis. And as you see from these data, at least half of the people felt there was a significant crisis. A significantly higher group, additively to nearly 90%, believe there is a crisis of some sort. And others were either uncertain or felt that no, this is not a true problem. So this slide indicates to me that scientists are concerned about this issue and are spending time thinking about it and are reacting to the sort of data that we see that suggests these problems exist. So these scientists were also asked whether they had established procedures for reproducibility, and the answers are as you see projected here. About a third of them said no, they really haven't focused on that issue yet. Another group of 26% say that they have procedures in place since they started working. Some have had procedures in place for a significant amount of time. And as a collective, this indicates to me that we have a fairly large group of researchers who haven't incorporated procedures or strategies to address reproducibility. And those that have indicate that their most popular strategy really is to repeat the lab procedures, therefore repeat what's been done in the past. And there was not a significant indication that new approaches, strategies, or training programs were being implemented. So when we think about the potential for a reproducibility crisis, what we're responding to are several papers and significant amounts of data that suggest that there really is a problem with either research credibility or research wastage. This paper is by economist Friedman, published in 2015, where he and his colleagues attempted to put to characterize the types of research that are being found to be not reproducible, but also to assign a cost to that problem. And as you see in this title, he indicates that irreproducible biology research costs are put at about $28 billion per year. And you will see in a later slide that that constitutes about half of the entire United States research budget. But they make an interesting point in this paper, suggesting, as you see quoted here, that the major cost of this irreproducibility does not lie with a faulty initial study, indicating that the science might be sound in many of these studies, but that wasted follow-up occurs, that is because there are false leads and problems with replicating the study, which also then results in delaying alternative discoveries due to the fact that resources are being spent on work that can't be reproduced. So this is more data from that paper, which, as you can see in the left column, $56 billion is attributed to the U.S. annual preclinical research expenditure. And of that total amount, you can see here that these investigators, through a review of many studies, classified 50% of those studies as irreproducible. And when you consider that this is just the United States research budget, and it includes private research institutions, academic research institutions, but if you consider that there's so much research being conducted outside the United States and internationally, so this, I think, indicates that we may have significant research credibility and wastage going on around the world. Now, to the right of this slide, these authors tried to, when they evaluated the research that they looked at, they tried to be able to categorize what was it in these studies that made them irreproducible by scientists that would come after them. And the four categories that you see there are how they were classified, the problems were classified. So well over a third of those papers could not be reproduced because there was not enough information related to critical biological reagents and reference materials. So the information needed to try to reproduce those results or that research was just not available. Nearly 30% had study designs that could not be replicated. 25% had faulty data analysis and reporting. So with what was in the paper, you could not reanalyze that data and get the same results. And then about 11% of the papers just did not provide enough information to allow for effective laboratory protocols to be created and used to replicate the work. So those categories of problems in research studies have been noticed by the United States National Institutes of Health, and they have been quick to respond to the studies that have shown that research replication does not meet our expectations and the type of data that Friedman just presented in that paper. And when you see the NIH in 2016 initiated a change in the application instructions specifically designed to try to improve research rigor and reproducibility. And so they are now challenging the grantees to specifically address the four categories you see on the slide here. The scientific premise of proposed research, they're asking grant writers to spend more time on that topic. They want a rigorous justification of the experimental design. They want clear consideration of sex and other relevant biological variables. And then this authentication of key biological and chemical resources should also be a familiar element based on the slide that we just viewed. Now, in spite of their efforts to request this kind of more vigorous material from grantees, they're also providing training opportunities on their website. But they very quickly and forcefully state that they know that the efforts that they are working hard on will not be sufficient to affect real change in what they call an unhealthy environment. So at this juncture, what the NIH and what other stakeholders too, it's important to realize that research stakeholders such as the funding agencies who fund our work and also the publishers who publish our work, and then of course the public, are also responding to the data that reflect problems with research reproducibility. This paper that I'm referring to, called Robust Research, Institutions Must Do Their Part for Reproducibility, is a call to action for institutions to look closely at what we're doing to support our scientists as they try to conduct good research. And I think this is a very interesting graphic as it shows the robust science kind of crumbling around the funders on the left, the publishers in the second column, and the poor beleaguered scientist who is trying to figure out where they fit in and how they can conduct the work they need to do, while the institutions are a little bit asleep at the wheel and maybe not responding as vigorously or robustly and certainly not as quickly as our funding agencies like the NIH and our journals like Nature and Science, which have already brought together teams of publications and tried to put in procedures to help support more reproducible research. So we're really being called on to conduct GIP, which in the article in the last slide refers to good institutional practices. Now, if you look at this slide, this shows the research arc of the researcher, which I introduced you to earlier, but then also the three columns indicate the types of courses that the Epigeum program is working to develop. So there's a transferable skills course, And then there's the research quality and reproducibility course, which we're talking about tonight. And then content related to professional skills of the mature scientist. But for me, what's interesting is when we talk about most of the research that's generated and the publications that we produce are being conducted by the staff scientists, the postdocs, and principal investigators are providing the mentorship and leadership. So a lot of this research is being conducted in this category of scientists. And so if we want to get at generating reproducible and publishable data and accurate research inferences, this is probably a wonderful target for us to focus on. And so I think we would all agree with this slide in that there are at least 3 pillars of scientific investigation. And of course, the ethics of scientific research is critical, and I would suggest that the ethics category has been addressed for some time now in terms of many institutional, national, international focus on the responsible conduct of research. So we typically in our institutions have fairly good training programs in the responsible conduct of research in terms of ethics and integrity. By that I mean the focus on fraud, plagiarism, and fabrication, and managing research budgets and all the sorts of rights and wrongs that we should do from the ethical point of view. But we may not be as strong in the area of our training in best practices for research quality and reproducibility. And I think it's true that funding agencies and publishers and our stakeholders unanimously agree that there is a need for some additional vigor and rigor in attention to pulling these 3 pillars together. And I think most of us would also agree that these qualities that we're looking for need to be discussed and encouraged among all members of the research community, including students, techs, assistants, and other staff. So these programs need to be integrated in the research context, in the research environment. There's a lot of conversation on this topic about what's needed. This quote that you see on this slide again comes from the robust research, institutions must do their part. And I think it is a good call to arms about what kinds of things we should be thinking of. This idea that many investigators determine whether trainees are ready to move on based on the number and impact factors of publications. And instead, should we be thinking more about whether our lab members really have a good, strong hold on research methods, study design, process, and particularly a standardized approach to best practices in scientific research. Now, in this paper, they suggest that institutional training should be compulsory and should ensure a common understanding of rigorous design, research standards, and objective evaluation of data. Now, I think there are pros and cons for compulsory institutional training, but I have no doubt that all of us would be better served by having effective institutional training around these topics. Now, this diagram, I know it's busy, but what I want to call your attention to, just briefly as the background, what you see are these 4 columns that illustrate where Epigeum is planning to provide course content along the arc of the researcher. Now, the course we're talking about tonight is the Research Quality and Reproducibility course, where the plan is to have courses in the development of your research project itself, the planning of that research, the important parts of executing quality research, and then communicating with stakeholders and writing good research reports. And so what I've shown on in the orange bars are where we're hoping that we're targeting the sorts of concerns that have been identified in the literature by people who are doing research on research and by funding organizations like the National Institutes of Health, who are saying, you know what, we think our scientists need a little bit more support in generating a sound research premise, in creating effective experimental design. Definitely improvements are needed in research management, monitoring, and documenting. And by doing all of that, we'll improve our research transparency. And then ideally, and I'm certain that this is true, that we would be better able to reconstruct our research data, which should contribute to improved research reproducibility. And then I also want to call attention to Epigeum's focus on mentoring at all of these levels, which I think is a very important feature, because in the past, our primary gatekeepers for research quality has been mentoring, and most of the time it's done very well, but we can all imagine a serious inconsistency when you consider all the individual mentors and all the individual scientists. So this is one way to bring some consistency and standardization of approaches to research quality and reproducibility. And this slide comes from the World Health Organization, who long time ago, in the early 2000s, published and made available a wonderful document called Quality Practices in Biomedical Research, where they really laid out a handbook for what kinds of things should we be standardizing, what are the best practices in conducting research. And in this table, they show us how sound scientific principles, which we rely on our scientists for every day, the good ideas, the great hypotheses, the strong study design, are absolutely required and are probably not so much affected. What am I trying to say? Excuse me. What I mean to say is we have the two contributors, the sound science, which is the role of the scientist, but then the good quality practices, which are some of the more mundane aspects, but absolutely critical in terms of how is the research managed, do we have good documentation, are we conducting our research with using questionable research practices, or are we being effective stewards of our data? And only when we have both of those qualities do we have credible research. So that basically sums up my vision for this course, is a way of taking best practices, helping combine that with the scientist's own understanding of their sound science, and bringing those two worlds together.
Thank you so much, Dr. Davies. That was really informative, and I think that Sarah's presentation is only going to add to that.
Thank you. I think mine will be fairly quick, but I am going to introduce how we're going to take those principles that Dr. Davies just laid out and propose a new training program that will hopefully help to address this reproducibility issue. So what you see here is a diagram. It's a simplified diagram, but this is what we call the research workflow. So you know, if you think of it from start to finish, it starts with kind of ideation and thinking about your research topic, what's a problem that you'd like to solve. There's, you know, the planning, developing protocols. Of course, there's funding aspects, compliance, actually conducting the experiments, and then, of course, the publication and data sharing. And so, as we saw in Dr. Davies' presentation, reproducibility can be compromised at any of these stages. You know, she made the case that, which I think is quite valid, which is that most reproducibility errors don't come from necessarily a faulty scientific idea or experiment, and are usually not the result of a lack of research integrity, although of course that's sometimes the case. But very often that problem comes in the data stage, the data management and data analysis stage, where sometimes a perfectly good study can be highly compromised by just a lack of training on data management, which is very complicated. And so this training program that we're suggesting to develop is, you know, is really for anybody who is involved in professional research. And this could be any, you know, postgraduate students, postdoctoral professionals, faculty, even senior faculty. It's not saying that they don't know how to do research, but just trying to inject a little bit of extra training and awareness of how things can possibly go wrong, and making sure that those studies end up with the visibility and the accuracy that they should. So I don't think I need to say too much more about the motivation behind Epigeum wanting to produce this program, since Dr. Davies covered that extremely well when she was talking about those studies and the reproducibility crisis. I'd like to emphasize that although a lot of our data and reports have come from the U.S., so these are a couple of quotes from Nature articles, and we've spoken about the National Institutes of Health. This really is a global issue, and I'm sure those of you in Australia will recognize this as something that's quite universal, as an issue that the scientific community really does need to address. So I think the statistic is that fewer than 10% of biomedical studies are in practice actually reproducible, and there's definitely a related issue in the social sciences, both of which rely very heavily on very rigorous data management in order to create a reproducible study. This RQR course that Epigeum is proposing has a couple of very wonderful lead advisors, one of which you've just heard from. The other is Dr. Richard Koiko, who is based in New York City at SUNY Downstate Medical. And so the both of them combined have fantastic expertise, and I've been working with them very closely on scoping out the content for this program, since I'm not an academic myself, but I've become very interested in this issue, and we don't believe that there's any existing training available that addresses reproducibility and quality in quite this robust way as what Epigeum is proposing, which is why I think this is really exciting and a very timely project. So, as I said, this is an online training program, and even though it says here it's for researchers in the early stages of their careers, as I said before, it definitely is not limited to early career researchers, although that's certainly the best place to start, if possible, if they can be introduced to RQR at that stage. That's kind of training them at the best possible time. But there are certainly many very experienced, very brilliant senior researchers who could benefit from a little bit of extra training and awareness in this area. So this is a very, very basic outline of the courses that we intend to include in the RQR program. And so, each of the bullet points is an individual course. And each course will consist of a number of modules, and I won't get too hung up on that terminology, but overall the entire RQR program is expected to be maybe 15 total hours of training, and it doesn't need to be taken in a linear fashion. It can be broken up and taken in any order that you like, but we were trying to design this program around that research workflow diagram that we showed at the beginning of my presentation, and start to address each stage of that research process with the framework of reproducibility in mind. So starting with, even coming up with your topic and solving a problem, applying for funding, all the way through the experimentation and communication and publication stage, to always think of those research skills in terms of RQR, which again we think is a fairly unique approach to research training, because again we're not necessarily introducing this as a program just to teach research skills, which I think a number of the intended users will already be quite familiar with how to conduct research, but just offering it in the context of quality assurance and reproducibility. So again, the core audience, which I said before, quite broad. The institution clearly, I think, benefits from having that peace of mind, knowing that their researchers, given that with major research universities, they rely very heavily on their research output as far as their reputation and being able to attract and retain talent, as far as the broader question of their contribution to the scientific discourse. So there's definitely a benefit to the institution as far as there. I guess the converse of that is, it helps to avoid the trouble that individuals and institutions can get into when they need to retract papers due to problems with reproducibility, and that can be a bit of a setback, it can be embarrassing, it can be a big waste of resources, and you saw that Dr. Davies had showed just how much of a cost there is to having this lack of reproducibility. So there is a cost concern, there's a social concern, there's a reputation concern, and the final bullet point here, which I like, is preparing the next generation of research leaders. And nobody knows what will come in the next few decades as far as the research environment and culture, but having this really great new generation of researchers who are very aware of not just the study they're doing at the time, but its ramifications as far as inspiring future research that can build upon it. That's really, really crucial. I wanted to just say quickly, before we go to the Q&A,
Sure.
So we are looking for collaborators for this program, and so just to briefly touch on that, so we're currently at the stage of signing up universities, mostly in US, UK, and Australia and New Zealand, to actually join us and help us to create this program together. And so that business model is that we share the costs involved, and so we would have between 10 and 20 individual universities who would join us to launch this program and help us to shape it in the best possible way. And that cost sharing is part of the program because this program is very ambitious and it will be expensive to produce. The benefit is that anyone who joins us in our collaboration will, once we publish it, receive access to the program in perpetuity, so won't have to subscribe to any license. So that's the basic business model. You can certainly get in touch if you'd like to know more about that, but this is what we're proposing, and we'd really like to hear from anybody who might be interested in joining us. Thank you.
Okay, fantastic. And just to keep everybody on the same page, right here, while we're doing the Q&A, here is the contact information for Sarah, myself, and Epigeum in general if you have any questions. Or you can follow the web address, epigeum.com/nextsteps, to learn more about our collaborations in general. While we were speaking, we did have one question come for Dr. Davies. Dr. Davies, as you mentioned before, the 2015 Nature article about the role of institutions was advocating for them to adopt good institutional practices. What type of practices were they recommending?
Okay, so that article really did provide some insight into what they're hoping for, and basically they noted that universities, as I mentioned, typically have programs that address research ethics, but they don't really have a process or what they call a standardized approach to address some of the questionable research practices or inconsistent mentoring that commonly occur. So there are several recommendations included things like in a standardized way, encourage more robust discussion of research methods in a formal way across departments. So instead of just talking within our own research groups, putting your proposals and your premises out there to sort of friendly external independent review within your university. They were also suggesting that we consider using non-punitive reporting systems to encourage that problems be discussed, so that incidents or errors or intriguing problems come out in the open and can be discussed and evaluated, looking for true root cause and unique strategies for addressing, so that everybody can learn from things that maybe go wrong. That's currently done all the time in mature quality assurance programs that have approaches, corrective action and preventive action approaches to non-conforming work. But they were suggesting that we consider that in the basic research setting. Then, of course, they focused on training and standards, and they, as I mentioned in my slide, they recommend that compulsory training in the areas of questionable research practices like study design, statistical approaches, bias. And I find this intriguing, and again, it's one of the things I like about the Epigeum model, and especially the consultative model, is that it's recommending that we really talk together about what we think a standardized approach or what best practices really are, and we commit to those as scientists. Another suggestion is that they really think it's time that we start thinking about records and quality management in the basic research setting. Again, standardized approaches to data record keeping and data management throughout the life cycle of data. And then, of course, they mention what many of us think about, which is, should we be reviewing the incentive system within the scientific community and the scientific reward system? And that, of course, is a very big topic that is worthy of conversation, but probably not something we can fix with a training program. I just had a couple comments as I listened to Sarah.
Sure.
that I'd like to make, if that's all right.
Of course.
One is that one of our dreams with this training program is that we will be providing, in addition to training content, we'll be providing tools and templates, some real practical things that our scientists can use to showcase the quality of their work or demonstrate the quality of their work. So we envision things like downloadable, again, in my world of quality assurance, we hope to be able to help them actually integrate a quality assurance program that they can then use to communicate with their granting associations or other scientists. So again, practical tools they can use to demonstrate the quality of their work. And then the second thing is, as I listened to Sarah talk about the collaboration groups, for me as a lead advisor, I see that as really critical to get these development partners in place, because it allows us to again have this consensus-based training where if we have 10 or 20 representatives from different universities around the world, well, we're going to come to agreement on what we think these best practices are and a best way to introduce them to scientists. And I see that as a very powerful platform, much more powerful than if I were to individually try to develop my own program according to my way of thinking. So I just wanted to add those two comments. Thank you.
Yeah, thank you, Rebecca. And I definitely agree. And based on our time constraints here, I didn't go into too much detail about that. But really, if anyone is interested in getting in touch with me, I can walk you through a lot more of the detail, because really the unique value of Epigeum really is in that collaboration model. It's very unique. We don't see a lot of other online training companies who do things this way, but as Rebecca said, it's going to be a much more powerful program having that collaboration in place and making sure that we're actually publishing something that is going to meet the needs of researchers who are actually doing this day to day. And even even with the fabulous expertise of Rebecca and Richard as our lead advisors, we really can't do that without the insights of the universities. And so please get in touch with me if you'd like to learn more.
Yes, and all of this information and contact information, again, is available on this last slide and also at epigeum.com. So again, thank you everyone for attending, and I hope you have a lovely day. Thank you, Sarah, and thank you, Rebecca, for participating.